In this video, we're going to go through the various food tests you'll need to know for your GCSE biology practicals. We'll be covering all the essential ones, including the Benedict's test, which is used to detect sugars, the Iodine test for identifying starch, the Biuret test, which checks for proteins, and the Sudan 3 test, which tests for lipids. If you're studying the Edexcel course, we'll also look at the Emulsion test for lipids. One key step that all these tests share is preparing your food sample. To start, take your food item and crush it using a mortar and pestle. Transfer the crushed sample into a beaker and add some distilled water. Use a glass rod to stir the mixture, helping some of the food to dissolve. Next, you'll need to filter the mixture using filter paper and a funnel to remove any solid particles. This leaves you with a liquid sample, ready for testing. Let's start with the test for sugars, which are the basic units that make up carbohydrates. Sugars come in two types, reducing and non-reducing sugars. Although you don't need to know the difference in detail, remember that the Benedict's test only detects reducing sugars. To carry out this test, prepare your sample as shown earlier and add 5 cubic centimeters of it into a test tube. You'll also need to set up a water bath at 75 degrees Celsius. Add around 10 drops of Benedict's solution to your test tube and place it in the water bath for 5 minutes. For safety, always use a test tube holder and keep the tube pointed away from yourself and others. If no reducing sugars are present, the solution remains blue. However, if there are reducing sugars, it will turn green, yellow, or brick red, depending on the sugar concentration. Now, let's move on to starch, which is another carbohydrate but much larger and more complex. Luckily, the test for starch is simpler. Just take 5 cubic centimeters of your sample and add a few drops of iodine solution. The iodine gives the solution a brown-orange color. Give it a gentle shake. If starch is present, the color changes to a blue-black. Now let's look at the Biuret test, which is used to identify proteins. For this, you only need 2 cubic centimeters of your sample in a test tube. Add 2 cubic centimeters of Biuret solution. It starts off blue. Give the tube a gentle shake. If protein is present, the solution will turn pink or purple. If you're taking the Edexcel course, you may be asked to use a more specific method. Add potassium hydroxide, followed by copper sulfate. Now for lipids, we use the Sudan 3 test. You'll need 5 cubic centimeters of your unfiltered sample in a test tube. Add 3 drops of Sudan 3 stain and gently shake the tube. If lipids are present, they will be stained red. Because lipids don't dissolve in water, they form a bright red layer on top. If you're on the Edexcel course, you might be required to perform the emulsion test instead. To do this, mix your food sample with 2 cubic centimeters of ethanol and shake it well. Then add an equal volume of distilled water. If lipids are present, the solution will turn a cloudy white. If no cloudiness appears, it means lipids aren't present.
If you haven't heard before, you can find all of our videos on our website geniushub.co.uk. Additionally, you'll also find MCQs, flashcards, past papers, and quizzes. Make sure to leave a like and subscribe for more content like this.